Good evening, all. Welcome to Chemflix. Hi guys, today we're going to discuss about bio-inorganic chemistry, which is very useful for HSST chemistry, PGT chemistry, CSIR, NET, GATE, UPPGT, AWES, and for all chemistry-based competitive exam based on a Pakka syllabus. So here, as we're going to discuss about the detailed syllabus of this bio-inorganic chemistry, we could find that metal ions present in biological systems. Metal ions present in biological systems. Then comes uh, classification of elements according to their action in biological systems. Then sodium potassium pump. You can expect questions from these areas: carbonic anhydrase and carboxypeptidase. Excess and deficiency of some trace elements, trace metals. Toxicity of metal ions, mainly mercury, lead, cadmium, arsenic. A reason for toxicity. Use of chelating agents in medicine. Iron and its application in biosystems. Hemoglobin, storage and transfer of iron. Actually, the syllabus I got it from somewhere over Kerala PSC, I think, detailed syllabus. So you can expect questions from this area mainly. Then this is also useful for PGT chemistry that we are discussing. A high level discussion happens here based on CSIR net previous questions. Then gate. Okay, so I think it's useful. Then this is part one of our video lesson, and here we're going to discuss about this metal ions present in biological systems. First point. Next one, classification of elements according to their action in biological system. Then sodium potassium pump we are discussing here, but we are not discussing carbonic anhydrase and carboxypeptidase, which comes under metalloenzyme, that we will discuss in the next lesson. Excess and deficiency of some trace metals, that also we are discussing here. Toxicity of metal ions, that also we are discussing here. Use of chelating agents in medicine, that also we are discussing here. Here iron and its application in biosystem, hemoglobin, storage and transfer of iron, that we are going to discuss in another one video. So hope you will love it and hope you will use it. Okay, utilize this video. Okay, so without wasting much time, we will directly move. The first topic is classification of elements according to their action in biological system. Of the 100 plus known elements, now you know that total 118 elements are there. 2 to 3 dozen, that means 2 to 3 dozen means 2 into 12 or 3 into 12, can be found in most organisms. Most are found in trace amounts, but 5 standouts. Which are the 5 standouts? Carbon, hydrogen, oxygen, nitrogen, and phosphorus. CHONP. You know that phosphate, adenosine triphosphate, biphosphate, diphosphate that you are studying, nitrogen in amino acids, then carbon, hydrogen, oxygen is essential for carbohydrates. Okay, so you know that. So these are the 5 standouts: carbon, hydrogen, oxygen, nitrogen, phosphorus. Carbon is in every organic compound. You know, organic chemistry is because of this carbon. Only one element, that is because of the carbon. It forms backbones of carbohydrates, proteins, lipids, and nucleic acids such as DNA. Carbon's unique ability to form 4 stable bonds, that is tetravalency, unique ability to form 4 stable bonds means it can form more molecular structures than any other atom. Hydrogen and oxygen bond as water play an essential role in living cells. We know that the importance of hydrogen and oxygen bond as water. Water, almost 70% of human body consists of water, right? So water is the primary solvent in which organic molecules are sustained in cells. Most biochemical reactions occur in the watery or aqueous phase. So most biochemical reactions occur in aqueous phase. Hydrogen and oxygen also play vital roles in producing energy in most cells. Carbon and hydrogen and oxygen are abundant in living matter. Nitrogen and phosphorus are more rare. Every protein has nitrogen in its molecular backbone. Phosphorus plays a key role in energy and information system of the cell. So these are basic idea of mainly about the standout elements, that is carbon, hydrogen, oxygen, nitrogen and phosphorus. Moving to the next, consider the content of the element in the body of an average healthy person weighing 70 kilograms. Oxygen, carbon, hydrogen, nitrogen, phosphorus. OCHNP. Oxygen 45.5 kg, carbon 12.6 kg, hydrogen 7, nitrogen 2.1 kg, phosphorus 0.7 kg. So the most abundant non-metal present in the human body is oxygen, that is almost 45.5 kg. Then metal distribution, first one goes to calcium. First place goes to calcium, 1050 gram. You know the importance of calcium. Calcium, where you can find calcium in bones and teeth and everywhere you can find calcium. That is almost 1050 gram. Then comes potassium, then sodium, magnesium, iron, zinc, copper, manganese, molybdenum, cobalt, and chromium. Data shows that abundance of copper, manganese from here, copper, manganese, molybdenum, cobalt, and chromium, etc. metal is less than 1.0 gram, but those are highly significant for the biological system. This partial data you can see here, it is 0.11, 0.02, 0.005, 0.003, 0.0014 gram. So this is less than 1.0 gram, but those are highly significant for the biological system. This partial data helps to predict the order of abundance of the metal ions in human body. It is showing a qualitative correlation with the order that no need. Now moving to the next slide. According to the action in biological process, and abundance of these elements can be classified in different categories based on the action in biological process and abundance. Essential elements or essential metals, trace elements or trace metals, non-essential elements or non-essential metals, toxic metals or toxic elements. So essential, oxygen, carbon, hydrogen, nitrogen, phosphorus, then comes sodium, potassium, pump. So sodium and potassium. Then comes magnesium, chlorine, calcium, sulfur, etc. These all are included under the category essential. Essential means they are absolutely necessary for running the life process. So essential means they are absolutely necessary for running the life processes. Then comes trace: iodine, iron, copper, zinc, manganese, cobalt, molybdenum. These are trace elements. They are essentially required for the life process but exist in low concentration. Then comes non-essential: aluminium, strontium, barium, zinc, tin, etc. Non-essential elements are not essential. If they are not present, other elements may serve their function. Then comes toxic: cadmium, lead, mercury. They are toxic elements and they are not required in the biological system. Then some metal dependent human system. Nerve, metal disvalent: sodium, potassium, magnesium, sodium, potassium, magnesium. Okay. Diseases: epilepsy, personality change. Okay, is there any disbalance or causes in sodium, potassium, and magnesium? It may cause epilepsy, personality change, etc. It will affect the nerve. And muscular system: sodium, potassium, iron, myotonia. Cardiovascular: heart, blood, magnesium, calcium, sodium. Metal disbalances: magnesium, calcium, sodium, hypertension. Then blood vessels: sodium, potassium, iron, copper, heart failure. Digestive: zinc, iron, liver cirrhosis. Copper, excess of copper results in Wilson disease. Previously asked a question. Copper, excess of copper results in uh, Wilson disease. Urinary: potassium, magnesium, calcium, renal insufficiency. Bone and skeleton: calcium, magnesium, osteoporosis. So these are some metal dependent human system. Then comes application of metal for medicinal use. So platinum is an anti-cancer agent. So platinum, it is in the +2 oxidation state. They are mostly widely used for the treatment of cancer. cis. It is in the cis form. Conformation, cis conformation. Cisplatin, carboplatin, everything is in cis conformation. Here, this one also in cis conformation. They can be used for the treatment of cancer. Other elements, other metals, you can see lithium. Lithium is for the treatment of depression, previously asked. Gadolinium for contrast agent. Barium sulfate, a contrast agent in radiography. Technetium for radiodiagnostic. Aurum for remission. Uh, SB antimony for eczema. Bismuth for gastric ulcer. Cadmium for carbohydrates. So these are the other metals and its application. Treatment of depression, which metal is used? Lithium metal is used. Contrast agent in NMR, which is the metal that is used as a contrast agent in NMR? Gadolinium. Which is the metal that is used for, which is the compound that is used for the contrast agent in radiography? That is barium sulfate. And for radio diagnostic of thyroid, which element is used? That is technetium, right? Now, descriptive study of some common metals in biological system. So we are moving. Calcium, first one about calcium. So about 99% of calcium exists in the human body as phosphate, resembling the mineral hydroxyapatite, that is C810PO46 OH twice in bones and teeth. The remaining amount found in cellular fluids exists in partly ionized or in protein bound form. Deficiency causes calcium deficiency. Calcium deficiency causes thinning and weakening of bones, important, osteoporosis, hypocalcemia. And the second one, important, excess of calcium leads to the formation of stones, hardening of arteries, cataract in the eyes. Calcium deposition causes hardening of arteries. Have you heard that, right? So excess of calcium leads to the formation of stones, hardening of arteries, cataracts in the eye, and the deficiency of calcium causes thinning and weakening of bones, osteoporosis, hypocalcemia. Then comes magnesium. Magnesium is an important pigment, chlorophyll pigment. In plants is a complex of magnesium with a porphyrin. It has good ability as a divalent cation to form octahedral geometry and can stabilize the structure without promoting energy loss by fluorescence. All enzymatic reactions in animals and human body are catalyzed by ATP. Magnesium acts as a cofactor of adenosine triphosphate. Number of biological processes such as oxidative phosphorylation, DNA transcription, RNA function, protein synthesis, and critical cell membrane functions are all require optimal concentration of magnesium. Important. Oxidative phosphorylation, DNA transcription, RNA function, protein synthesis, and critical cell membrane function are all require optimal concentration of magnesium. About 99% of magnesium in human body exists in intracellular regions and bonds. Deficiency, important. It causes hyperexcitability, dizziness, muscle cramps, muscle weakness, fatigue. And excess of magnesium causes hypermagnesemia, which occurs rarely because the kidney is very effective in excreting the excess magnesium. Next is about potassium. So 90% of potassium in human being found as both intracellular and exchangeable, that we are going to see in sodium potassium pump. It is reason for sizable potential difference across cell membrane, which is responsible for neuronal signaling. Hypokalemia may result deficiency due to potassium. Then comes sodium. Measure part 65% of sodium exists in human as extracellular cation. That also we are going to discuss while we are discussing of sodium potassium pump. And excess results in blood pressure, that is hypertension. Alkali metal ions like sodium, potassium have highest ionic mobility in biological media that make them vital for human body because they provide very quick response in biological action. Then comes cobalt. It is a metal center of vitamin B12, that is cobalamin. Promotes RBC formation, activates some enzymes. Excess can result in vomiting, nausea, vision problem, heart problem, thyroid damage. Then comes zinc. Deficiency of zinc can lead to dwarfism, sexual immaturity, diarrhea, and pneumonia. Largest amount occur in liver and bone. Catalyzes the peptide hydrolysis, that is carboxypeptidase, that we are going to discuss in the next video. Carboxypeptidase, it is a metal enzyme and the metal is zinc. Then comes molybdenum. It is an abundant element that probably used by all organisms as a redox catalyst for the transfer of oxygen atom derived from water. It plays intriguing role in nitrogen fixation. So molybdenum is useful in nitrogen fixation and important a part of iron sulfur protein deficiency. Delays seizures, visual alteration, and neurological changes. Molybdenum acts as an active site of nitrogenous bacteria along with the iron. Then comes chromium. Chromium is a part of GTF, that is glucose tolerance factor, which includes one CR3 plus and provide aid in insulin binding to site. It helps in lowering of cholesterol and triglyceride levels. So glucose is there, cholesterol is there, triglyceride is there. Everything is related to chromium. In excess amount can be quite toxic. It is known as carcinogen and toxic metal. It can result in skin and lung cancer. Then comes copper. Copper, liver, kidney, heart, and brain in concentration. The enzyme tyrosinase is important. The enzyme tyrosinase catalyzes melanin synthesis in melanosomes and require copper for its activity. Copper may be involved in free radical production via Haber-Weiss reaction that results in mitochondrial damage, DNA breakage, and neuronal injury. Excess also can result in Wilson's disease that we already discussed. So it is involved in the free radical production by Haber-Weiss reaction that result in mitochondrial damage, DNA breakage, and neuronal injury. Deficiency can result anemia because it is important part of ceruloplasmin which has a role for iron storage in the body. So copper deficiency creates anemia because it is an important part of ceruloplasmin which has a role for iron storage in the body. It is believed that it is involved in the process of oxidation of iron to ferrous to ferric to transfer the iron from ferritin to transferrin. Then comes iron. About 60 to 70% of the total amount of iron in the body present in hemoglobin in red blood cells. 3 to 5% is in muscle myoglobin. 15% is bound to ferric iron storage cellular protein that is ferritin, and 0.2% occurs in the component of critical respiratory enzymes, and 0.04% is bound to serum transport protein transferrin. Iron deficiency causes anemia because hemoglobin has iron as important constituent and excess can result in siderosis. Excess amount of iron can cause siderosis. Fe3 plus help in coagulation of blood and seal the blood vessels. So Fe3 plus, Fe in 3 plus, that is ferric helps in coagulation of blood and seal the blood vessel, found in nitrogenous genase along with the molybdenum. Iron along with the copper work as electron transport device in biological system, mainly in plants. Iron electron transport, that is root redoxin, 4FeS, plastocyanin, like that we will discuss. Then comes platinum, bismuth, ruthenium, aurum. These elements have not known for deliberate biological function. Those diasodium, gold. You know that gold is very important for treating osteoporosis, right? Arthritis, like so. Diasodium, gold, one thiomalate, myocrystin is used for arthritis. Platinum is even more non-prevalent for biological systems. So platinum is non-prevalent for biological system. Next, we are going to discuss about toxic metals, that is mercury, cadmium, lead. You know mercury, Minamata disease, cadmium, Itha or Och disease, lead. Lead poisoning can lead to anemia because it inhibits the activity of ceruloplasmin enzyme that gets involved in the synthesis of heme, that we already discussed, right? Iron, cobalt, like lead also, like mercury, cadmium, lead inhibits SH enzyme but less strongly. Okay, moving to mercury. Minamata disease, the main cause is mercury. Organic mercury compound, which are fat soluble, can cross the blood-brain barrier and can result in neurological damage. Toxicity of mercury is based on its strong affinity to thiol of cysteine residue. Same thing everywhere. Mercury is a soft acid and thiol group is a soft base, so strong interaction occurs between mercury and thiol group. Then comes cadmium poisoning leads to nausea, salivation, vomiting, diarrhea, abdominal pain. Cadmium ion is similar to zinc in size and charge, so it can be replaced like in the place of zinc, cadmium gets deposited and toxicity may happen. Then carbonic anhydrase, dipeptidase, carboxypeptidase, etc. Then cadmium, like other toxic metals, affect the active conformation of biomolecules due to strong binding. Then detoxification of the chelating agent in medicine. How you can treat the metal poisoning. That is what is called detoxification. Detoxification, metal poisoning. Metal poisoning means if there are any mercury poisoning, if there are any cadmium poisoning or lead poisoning, how you're going to treat. That is what is called detoxification. And chelating agent. What is the role of chelating agents in medicine? So mostly the treatment of metal poisoning, that is detoxification, is done by using chelating agents. It is hoped that the chelating agent will form soluble, stable and non-toxic complexes which will be readily excreted. There are following criteria for designing a potent chelating drug. The chelating drug should bind the toxic metal in such a way that it can compete with the biological ligand. It should be selective. It should not go in metabolic process. It should have proper approachability. It should form non-toxic complex that should be easily excreted from the body. So these are the main things that you have to keep in mind while you are going to design a potent chelating drug. It must bind the toxic metal. It should selective for toxic metal. Then it should not go in metabolic process. It should have proper approachability at specific site, and it should form non-toxic complexes. Some of the examples of chelating agents are EDTA, penicillamine, etc. EDTA, that is ethylene diamine tetracetate. Nickel EDTA complex was used for the treatment of breast cancer. Calcium sodium EDTA, calcium disodium EDTA is used for lead poisoning. Cobalt EDTA is used for cyanide poisoning. And D-penicillamine is used to remove the toxicity of copper. Okay, now comes the sodium potassium pump and ionophores. This is the last topic of this session. Sodium potassium pump is to maintain the cell potential across the cell membrane. So what is the main thing? To maintain the cell potential across the cell membrane, cell must keep a low concentration of sodium ion. So low concentration of this is cell. Cell must keep a low concentration of sodium ion. So here sodium ion is very low. That means potassium ion is high. And high levels of potassium ion within the cell. Outside the cell, there are high concentration of sodium, exact opposite, a high concentration of sodium ion and low concentration of potassium ion. So what happens? Some gradient is created among these inside and outside the cell, right? So outside cell, there are high concentration of sodium and low concentration of potassium ion. So diffusion occurs through ion channels in the plasma membrane. So some ion channels are there. Through this, diffusion occurs. In order to keep the appropriate concentration, the sodium potassium pump pumps sodium ion out from here to here. It pumps sodium ion out and potassium ion in. So potassium ion in through active transport. The ionic transport conducted by sodium pump creates both an electrical and chemical gradient across the cell membrane. Which enzyme is used? Sodium potassium ATPase is the major component of sodium potassium pump, which is essential in creating membrane potential. This intrinsic membrane protein consists of two components, that is a 100 kD catalytic subunit and a 45 kD associated glycoprotein organized into an alpha 2 beta 2 tetramer that we will see now. So this is actually happening here. This is the mechanism. Outside here, what happens? Na plus will 3 Na plus will comes inside, and first the sodium potassium pump bound with ATP binds 3 intracellular sodium ion. Then phosphorylation of an ASP residue, ADP residue occurs, which provide the conformational change and weakens the binding of sodium ion. here, right? ATP, ADP, it weakens the binding. And the a conformational change in the pump exposes sodium ion to outside, less sodium ion outside and released. After this, ATP is hydrolyzed during this process, with the release of ADP and change in conformation again occurs that helps to bind two extracellular potassium ion. So two extracellular potassium ion, they are binding. Then potassium binding leads to dephosphorylation. So dephosphorylation, actually here phosphorase here. Potassium binding time, dephosphorylation to original conformation. And in this conformation, ATP binds the pump, reorients and release K plus ion inside the cell. So this is actually happening. This is the mechanism of sodium potassium pump. And the last point that we are going to discuss, that is ionophores. Ionophores means they are biological entities which help in transport system of ions, manganese, potassium, etc. across the cell membrane. So these are biological entities which helps in transport the system of ion, transporting ions. Their exterior boundaries become a lipophilic in nature, while interior region have binding capacity for metal ions, for example. Okay, so exterior lipophilic and interior this is lipophilic, and interior they have a binding capacity for metal. Example, valinomycin. Valinomycin, I think previously asked question. Nanosine, gramicidine, etc. Okay, so that's all for today. Uh, if you found this video helpful, then please click on the like button and also please subscribe to my channel. So thank you once again.